Yeah.
Okay.
Okay. Oh, let's see what I'm saying.
It's okay.
Okay. Anyways, welcome to the skill seminar. Thank you all for coming. Today we have Kailey, who is a fifth year CCB student in Neil Tsutsui's lab. And she's passionate about both evolutionary dynamics in social systems and science communication. And she received her Bachelor's of Science at the University of Rochester in computational biology, has been a member of the Tsutsui lab for the last 2 years. Thesis work involves investigating the evolutionary processes with unicolonial ant colonies, and specifically how worker evolution can be impacted by colony structure in different ant species. She's also interested in the long-term evolution of such systems and has used SLiM extensively to investigate how spatial dynamics within supercolonies can affect how these groups evolve over time. Really excited to have her.
Thank you.
Yeah.
Excited to be here. All right. Hope we're all ready to learn about some SLiM. So SLiM is a forward genetic simulator. I don't know what the acronym actually stands for, but it's an evolutionary simulation framework that basically emphasizes flexibility. So it has several advantages that they cite, just to give you a brief overview, namely flexibility. So if you've even taken a look at the manual, it is almost 900 pages, which should speak to how flexible this simulator truly is. It's essentially, they developed their own scripting language for it called IDOS. So anything I've ever been needed SLiM for, I've been able to kind of create a function for, or create sort of a biologically realistic model for. So really the only limit is your imagination with this simulator. So that's kind of its main advantage, with the large overhead of having to learn of how it works. So hence you being here and learning kind of the ropes for it. It performs relatively well as long as your population sizes stay within a reasonable limit. It kind of depends on what you're doing, but it tracks every single mutation and every single genome of each individual. And so it kind of scales with how many mutations you're inputting and your size. But I found that the performance is relatively good. I don't really have any frames of comparison for that, but it's not debilitatingly slow. And then there also is a really helpful interactive GUI, which I'll be kind of going over today as well, as I'm doing some of the scenarios that I'll go through. And the GUI just has a ton of really helpful features in it that will really help you debug stuff very quickly, and also figure out kind of step by step what's going on in each generation. So I'll go through some of the features of that as well. So you can find SLiM. Hopefully everyone's already done this or already has the GUI and SLiM on their computers. If you have a Mac, which it looks like everyone does in this room, Deanna, I don't know if you do, but it's really easy to install. You just hit the Mac OS installer and it brings you through the whole thing, and it installs SLiM as well as the GUI. So that's how you would get there. It's Simon Messer put this out along with Benjamin Haller. So it's on the Messer Lab website under SLiM software, presumably. And then that's Windows machines if anyone's interested. So the kind of first main distinction that is important with using SLiM is if you want to be simulating a Wright-Fisher model or a non-Wright-Fisher model. And so they differ in kind of the structure of how each tick works, the ordering of events, and also a lot of the housekeeping things that goes on during the generations. So in general, I typically use the non-Wright-Fisher models when I want kind of more biologically real scenarios. So it kind of increases your realism, with the caveat of it being kind of, you have to manually housekeep a lot of things that are taken care of for you in the Wright-Fisher version. So I won't go through every single difference here, but essentially the main differences between the two types are in the Wright-Fisher, fitness is calculated a little differently, and I'll go into specifically kind of those differences at the very end. But Wright-Fisher tends to deal with kind of more complicated and niche cases of things. So you kind of just have to go through the manual and look, but it's basically the fitness is either impacting your size of your population in the non-Wright-Fisher case, or that's taken care of for you in the Wright-Fisher case, where you just have to think about kind of the logistics of your simulation a little bit less. So yeah, I'll kind of go through. But Wright-Fisher is the default mode, so you have to specify if you want your model to be non-Wright-Fisher. But again, the non-Wright-Fisher model is used to kind of model various different advanced scenarios. So I'll go through some of those if hopefully we reach them by the end. I have a few of those scenarios for you. But in general, the non-Wright Fisher models are more individual based. They're more explicit. So you have to kind of explicitly account for like fitness per individual. You have to explicitly control the size of your population in a precise way. They tend to have kind of more biologically realistic dynamics because you can just have kind of more control over different aspects of fitness, reproduction, mate finding, that kind of thing. But they, also tend to be more complex because of that. And again, Wright Fisher is the default mode because it's simpler, and SLiM just takes care of a lot of that for you. Here are the ways that the different models vary. So I won't go through everything here, but Wright Fisher has non-overlapping generations. Non-Wright Fisher, you can have overlapping generations where you have different generations of reproductives in the same simulation. And it varies in all of these other ways as well. But mostly I'll be going through Wright Fisher examples, but just wanted to make you aware of the differences there a little bit. So just a conceptual overview of kind of how things are built in SLiM and how kind of the bare bones model runs. So basically, it focuses on simulating individuals, which each by default have 2 genomes. So I also have highlighted here any text that is blue is the name of a class in SLiM. So there is an individual class, there is a genome class, and all of the classes in SLiM obviously have different methods. And so all of those are outlined at the very end of the manual. But just to give you an idea of how you can start calling methods and kind of what's possible on the individual level versus the genome level. So you can change the ploidy of your individuals. You can do haplodiploid, you can do haploid, you can do diploid. I don't know if you can do triploid. I haven't looked into that, but it's diploid by default. So thus each have 2 genomes. So the individuals here are represented by the yellow squares, and each of these is just a chromosome. And essentially what the chromosome by default is it has some set length L that you specify. And then each of the squares is just an empty bin. And so unless you ask SLiM to explicitly model nucleotides, it doesn't by default. It just stores if there is a mutation object there or if there is not. So each of these bins is just like a potential for a mutation to occur there. Okay, so mutations and substitutions, this is how it works in SLiM. So each instance of a mutation in various different individuals are basically just references to the same mutation object, which makes sense and kind of helps with efficiency here. So again, mutation, because it's blue, is a class as well. So all the individuals containing a blue mutation here in this particular bucket, this blue state is just references to the blue mutation object. And that mutation object has various different features. So it has a base position where it occurs on your chromosome, it has a selective coefficient, and it has a dominance coefficient as well. So all of these are just reference to the same blue mutation object. And another thing too is you can have stacked mutations in SLiM. So you can have 2 mutations occurring at the exact same base position. Each mutation will refer to a separate mutation object, but you can turn that off as well if you want to. Okay. So when a mutation becomes fixed in a population, it no longer is classified as a mutation object. It converts to a substitution object. So basically this blue mutation now, since every individual has it, will now become a new type of object, substitution. Again, this is done mainly for efficiency purposes because it no longer is factored into any sort of fitness calculations that are done per individual per generation. So it's just easier for SLiM to not have to account for all of these mutations in every individual. So it is no longer affecting fitness, which makes kind of intuitive sense because if everyone has it, you're not going to be different by having a mutation. So it no longer gets factored into the fitness calculation at the end. So you can also turn that off if you want to. Yeah. You can what? Yes, you can. Yeah, you can also turn that behavior off, but in any bin, you can have multiple mutations if you want to. Usually that's like not a super desirable behavior because it's not necessarily reflecting biology, but you can do that. Yeah. So yeah, the mutation gets removed, but yeah. So then when you're building up your chromosome, the chromosome again is another class, and it contains these various different what are called genomic elements. So this is what the class is called in SLiM, genomic element, camel case, which each have a particular type. So you specify the genomic element type, and the type is mainly distinguished by the mutations that are allowed to occur in it. So the point of this is basically to explicitly model like different types of elements that you might want on your chromosome. So say you want non-coding regions and also exons and also introns. You would specify the types of those different genomic elements at a particular spot in your genome. Say you want to model a particular gene or something that you know of, and you know kind of the general length and its proximity to other types of elements. You can model all of those and specify them. And again, the main distinction between the element types here, at least according to the eyes of SLiM, is just what types of mutations can occur in those elements. So an example here is in a non-coding region, you probably would only allow for neutral mutations to occur in that region. Whereas an exon, you would allow all 3 types of mutations to occur. And that is explicitly specified in the setup when you initialize your genomic elements on your chromosome. You can also just have the entire thing be 1, but you have to specify your genomic element type. So you can't just say like, oh, I don't want a type. You have to actually explicitly say that this type will be the entire chromosome and it will allow all mutations in the simulation to occur there. Okay. Questions, anyone? Good. Okay, the last thing I'll go over is migration and subpopulations. So subpopulation, again, is a class. And so you always have to initialize subpopulations in SLiM, even if you just have one. You have to initialize it and say like how many individuals you want. And so this is basically to easily model things like gene flow or migration between subpopulations. So in SLiM, mating only occurs within a given subpopulation. So if you include 2 or more, mating will not occur between those unless you explicitly designate migrants to be exchanged between those subpopulations. So again, you have kind of an innate setup for reproductive isolation within SLiM by initializing different subpopulations. And so this, again, allows you to easily model gene flow. This is an example of like a stepping stone river system, which I thought was pretty cool. So you have like 10 different subpopulations, and you have each of these exchanging migrants where one direction is kind of a lot stronger than the other. And this is, if you're trying to model a particular river system or something where you'd expect migrants to be flowing between subpopulations in a particular direction, you can easily do that. Okay. And then also if we add in spatial layers, so SLiM has the capacity now to use spatial layers. So you can do 2 or 3 dimensions if you want. And so you can also easily model spatial competition or spatial mate choice preference using the idea of subpopulations, because your spatial competition or mate preference or whatever it may be will only occur. It's basically a method of the subpopulation that you're calling. So it will only occur within that particular subpopulation. So you can specify different dynamics going on between different subpopulations, which is, I have an example at the very end. I don't know if we'll get to it, but okay, yeah, that wasn't the last thing. This is the last thing. So recombination and gene conversion. So by default, SLiM operates on a crossover breakpoints model for recombination, which is realistic enough. They kind of go through this distinction of crossover breakpoints versus double strand break model, which is just kind of a more complicated version where you can model gene conversion. And I won't get into the details there. If you're interested in modeling that particular type of recombination, it has more biological realism for those purposes. But for most purposes, the crossover breakpoints model works just fine. So basically just selects the number of breakpoints that you want to draw based on your designated recombination rate. It selects the locations randomly based on that rate, and then it flips your genomes accordingly, and it produces this recombined genome. So as you would expect kind of it to work. Okay, and then the last thing I wanted to kind of go over is how fitness is calculated. So you have your populations of individuals, and then in order for evolutionary dynamics to occur, you want some to have higher or lower fitness so that you get evolution, right? So the calculation of fitness or the definitions vary between your different model types. So jumping back to the distinction between right Fisher and non-right Fisher, This is kind of like the main important difference that you need to remember if you're going to decide if you want to choose one or the other. In Wright-Fisher models, what you have is called like a relative fitness. So fitness for everyone is kind of scaled so that the population size is always relatively constant, and that size is set initially by the model. So you don't really have explicit control over higher fitness leading to like higher population sizes or extinction happening in your simulation, for example. And fitness here is defined as the probability that an individual will reproduce in the next generation. And so it's like a subtle but important difference in non-Wright-Fisher models, where fitness is defined as the probability that an individual survives to reproduction. And then reproduction is more or less guaranteed unless you designate it otherwise in a non-Wright-Fisher model. And in a non-Wright-Fisher model, you have absolute fitness and not relative. So basically, it actively influences the size of your populations, which is very important and can kind of lead to a lot of headaches if you don't like correct for it and think about it ahead of time, because it's really easy to drive your populations to extinction or have them have exponential growth and then you get a segfault and it just doesn't work. So you kind of have to really explicitly think about how your mutations are influencing the overall distribution of your fitnesses for all of your individuals in your population. So 1 being you're guaranteed to survive to the next generation, 0 being you're guaranteed to die before you reach the next generation. But if you induce a lot of mutations that are beneficial, for example, your distribution might even, the mean might even go past 1. And so every individual then survives, and then you have kind of an exponential case where you have this unchecked growth. So you really have to think about how to control for your sizes when you're defining and modifying fitness in the non-Wright-Fisher case. But yeah, and then this is how fitness is calculated. So it's calculated multiplicatively for every individual, and it's on a mutation by mutation basis. So it's just 1 plus your selection coefficient if you're a homozygous, or 1 plus the selection coefficient weighted by the dominance if you're heterozygous. And this happens for every single mutation on your individual genome. And that's how fitness is then calculated. And then some random number is drawn from 0 to 1 in SLiM. And if you're under that random number, you die. And if you're over it, you survive. All right. So that concludes the slides portion of this talk. Anyone have questions about anything like that? It's all kind of basic stuff. Okay. So I am now going to attempt to all right. I don't know what this is. Okay. I'm going to save that. All right. So bear with me. Oh, should have saved that. Well, I just kind of arrange things. So for this portion, I have a bunch of basically just scenarios that range from very introductory to pretty advanced and span Wright-Fisher versus non-Wright-Fisher as well. So that's what we'll be doing for the rest of the talk. Okay, so again, if you have questions about anything I'm doing, just interrupt me and ask. It's all good. And we will get started with this part. So before I introduce the first scenario, which will just be your basic neutral simulation scenario, I just want to show you a few of the features of SLiM. So this is what the GUI looks like. If you want to follow along to any of the scenarios that I'll be going over, they're actually all pre-downloaded when you get the GUI. And so you would just go to File, Open Recipe, and the recipes are laid out in order of the chapters in the manual. And then each one has your subsections of different ones. So you can usually find whatever you are looking for and then modify that recipe, which is a really nice feature of the GUI. And I didn't even learn about this until a few months ago. I was manually writing my own. But yeah, this is like a very good starting point if you're not super familiar with like every single how to model like a kind of advanced scenario. So if you do want to follow along right now, you can also just use the recipes that are laid out in here. Okay, so I'll just like take you through the main things about the GUI. So you'll see that basically I'll just run this simulation so you can kind of see like what output looks like. But you have your main code block here, and then you have a bunch of really nice features. So these all are basically, these are for like error checking if you want to check to see if it will run before you even try to run it. This is pretty printing. So if you're copying and pasting code from a different source and your spaces and indentation are all messed up or something, this will just immediately correct it for you. There are a few others. This is like a help script. This is like, it opens an IDOS console. So IDOS is the scripting language that was developed for SLiM, and that's what you'll be scripting in. So if you want to do any sort of scripting that doesn't explicitly involve like a SLiM function or method or something, then you can consult the IDOS manual and see if it'll work in there. This is where your output will occur if you are running something that generates some sort of text output. You don't always have to do that, or it could just output a file for you, which is what I always do. But if you want to just like visualize something explicitly, this is where it will end up. This is your simulated chromosome. So each of these bars is a mutation. And so we specified in this very basic intro simulation that we want our chromosome to be 900 or 99,999 base pairs. And another note about this too is that this basically constitutes every single mutation in every single individual that is in the simulation right now. So it will be an aggregate of everything going on. The color of the bars indicates the relative selection coefficient of each mutation. So yellow means neutral in this case. And that's why each of these blocks up here is an individual. And so they are also colored by their relative fitness. So yellow will always mean neutral. Veering towards red means deleterious, pretty intuitive. And then if they're starting to be colored green, that means you have some adaptive mutations going on, or their fitness is higher than like maybe 0.5 or something, or whatever kind of the mean is. This is just information about various different subpopulations that you might have. So some bookkeeping here about selfing rate, number, sex, if you're initializing that. And then this is how you run your simulations. So each new run, if you want to start over and run it again, you hit recycle and then hit play to run the entire thing to completion. But if you just want to run it tick by tick, you would hit this little button here. And then the speed of which it runs is modulated by this slider. So if you actually want to look at the dynamics happening at a slower pace, you can just change it, and this will run now a lot slower, and you can do it fast. Okay, so that's the main parts of the GUI. So I'll get into now starting with the basic simulation part. So I'll start with a basic neutral simulation, and I'm just going to do this kind of block by block so you don't get ahead of yourself. But I'm starting at the introduction in case somebody has like never used SLiM before, so there's probably a lot of review for a lot of people. But you're always going to start your simulation with an initialization block, and this is where you're going to have your required initialization methods. So you always have to initialize some mutation rate. The default will always be 1 times 10 to the -7. You can make this whatever you want. This is where you start to initialize your mutations. So initialize mutation type, basically, yeah, like it says, it initializes the type of mutations that you want. So for this particular mutation, we want to call it M1. So whenever you're referencing it in your simulation, you can always reference it by its name. This is its dominance coefficient for heterozygotes. And then this parameter is F refers to fixed, and it refers to the selection coefficient we want for this mutation. So for a neutral mutation, we want our selection coefficient obviously to be 0. You can also model, obviously beneficial or deleterious mutations, and you can keep the selection parameter as fixed if you want, but you can also make it a little fancier and draw your selection coefficients from any particular distribution that you want. So I usually use exponential for my beneficial mutations and a gamma distribution for my deleterious mutations. And so then you would specify the parameters of that distribution to the right of your flag. So that would either change to like E or G or N if you want Gaussian. Yeah. Yes, exactly. So every mutation that will occur will always have the exact same selection. But if you wanted to make it a little more realistic, there's always ways to kind of like introduce complexity to it. So that would be how you change that. Yeah, other questions? Okay. So then this is, you're introducing your genomic element and your genomic element type. So this is a really simple mutation or a simple simulation here. So we are just initializing that the entire chromosome is one type of element, which is why it's all blue. If it were different, it would color them differently on this bar up here. But you always have to initialize at least one genomic element and then specify its type. So we'll just start with this one first. So we're initializing this one genomic element. We're going to call it G1. And then here is its range. So you specify the start and the stop. And so this is just specifying how long you want your chromosome to be. And then you have to specify its type, like I said. So the point of doing initialized genomic element type is basically just to tell SLiM what mutations they can put there, or it can put there. So that's all it is, but you have to specify that. So here we're saying on G1, which we've defined below, we want M1 to occur. And then to the right is the proportions or the rates at which mutations are placed. So let's say you want to model like some realistic dynamics between beneficial and deleterious. You would include a vector instead. So vectors in IDOS are C, kind of like R, where it's C and then parentheses, and then you would do a comma separated list of your mutation types. Let's say M1 is deleterious, M2 is beneficial. And then the same goes for your selection or your ratios at which you want these mutations to occur. So if we wanted to model realistic dynamics, maybe it's like, I don't know, 10 or 20 to 1 or something like that. So it would be, these are all relative ratios as well. So you can literally just type 10, 1. And this will just take these numbers and generate a ratio for you. So this could be 20 to 2 or 2000, whatever you want. It doesn't actually have to be scaled to 1. Okay, so let's get rid of that because there's no such thing as M2 in this simulation. Okay, so now you initialize your element type. The only other thing to do is initialize your recombination rate. By default, it's always 1 times 10 to the negative 8, but you can do this whatever number you want. You can also introduce your own recombination maps. So if you have like a map for your organism that you can input it into the simulation. You can read it in as a file, or you can kind of manually hard code that in. I don't have an example of how to do that, but it's possible if you wanted to add more complexity to the recombination rate. And then it will kind of follow those dynamics across your whole chromosome. Okay, so yeah, we already ran this example of the neutral simulation, so I don't really need to go over it again, but that's how it works. So also for SLiM, usually you have what are called event types. And so you're required to have one early event at the very least, and one event either early or late that basically kills or ends your simulation. So here we have an early event that we're defining. So the one next to early means that we won't only want this event to occur once in the first generation of our simulation. So in this early event, we are now asking SLiM to call on the simulation class and add our subpopulation. We're going to call it P1, and then we're defining our subpopulation to have 500 individuals in it. And by default, these will be sexless hermaphroditic individuals, and they will be diploid. Okay, and then we're not doing anything in this simulation that is interesting. We're just defining our subpopulation, but I'm just showing you minimally what needs to be there. And then in our late population here, we have our last generation that we want it to run to. So this will run only once in our last generation. And this particular call, there's lots of different ways to generate output in SLiM. One of them is sim.output full. So this will signal to SLiM that we want the simulation to end, and we want it to output the full record of everything that went on. So I won't go through this in detail, but essentially it records information about each mutation that occurred in your simulation. And so all of these columns have some meaning. You'd have to like go in and text edit this if you wanted to use this information. So there are easier ways to generate output that you can use in downstream tools if you want. But this is just kind of like a bare bones way to generate output. You can also, it also shows you information about each genome that occurs, okay, it's just repeating over and over. There's other information too, geez. Oh, okay. Let's just change this back so I can show you. Okay. It's not happy. All right, so yeah, it's also helpful because it debugs it for you like that. Okay, yeah, so there's information about lots of different things, and there's a lot of mutations that occurred here, so that's why this list is super long. But yeah, also information about your individuals, right? If you want to output that. Again, this is just one of many ways. So a few different ways to generate output in SLiM. You can generate a random sample. So this is what it would look like if you wanted to do that instead of information about your entire population. So this is if you were to model like what your dynamics would look like if you only have a sample size of like 10 that you gathered from the field or something. So you can output a random sample. This is a method on the subpopulation class. So you're calling it from key 1 and you're saying, I want to sample 10 random genomes. There's another way that you can sample individuals instead. I don't actually know what the command is, but it's probably like output individual or something. But this is how you would generate a sample. And then, oh yeah, okay, I didn't go here. I was thinking ahead. So this is how you would do it for individuals instead of genomes. So basically, you first sample a random subset of your individuals in your subpopulation. So first you're getting the vector that contains all of your individuals. So again, this is a class and you're calling it all individuals. And then you are basically outputting a sample of all of your individuals, or you're first gathering a random sample and you're weighting it by some parameter based on the mutations they contain. And then you can call the output method on the genomes class or the genomes, class, which is also, you can call that on the individual class. Okay, so then that would generate output for each individual instead of each genome. But yeah, there's a bunch of different varieties. You can also output VCFs, which I use all the time, especially for like downstream tools that use VCFs as input. There's MS, there's a lot of stuff. So all of those details are obviously in the manual if you're interested, but just know that there's a lot of different ways you can output things. Okay, so then the next one I'll go through is simulating gene flow. So this is just like a very basic addition to our model where we're still having a neutral simulation, but we're adding some demographics into here. So the same initialization block up here, we have a mutation rate. We're initializing one mutation. It's neutral. We're specifying its selection coefficient as 0. A single genomic element, like the same parameters as before. And then we call an early event here. So early and late events are where you're going to be coding your basically anything that doesn't have to explicitly do with reproduction or fitness modification. Any kind of general scripting, like you're adding a subpopulation, you're, coding your own function or something like that, you generally would script them in early or late blocks because if you're trying to script something in, I'll show you later, but there's certain function calls where you can't just input regular code. Yeah, I don't want to confuse you all, but basically we're scripting this in an early block. And so early in the Wright-Fisher context means before your reproduction event. So before any reproduction is occurring, we want to create our subpopulations. So in this case, we're creating 5 subpopulations. And then this is how a loop is structured in IDOS. So it's, I don't know what it's similar to, maybe R or something. But you just do 4 and then parentheses is your iteration. And then you just indent, right? And then sim.add subpop is how you add a subpopulation, which we saw before. And then we're doing a nested loop here because we basically, in this example, want to create a metapopulation where you have migration occurring between every single possible pair of subpopulations in your simulation. So this is an easy way to do that. We're including this clause in an if statement because we don't want migration to occur within itself. That's illegal in SLiM. But basically, we want every possible pairing, hence the nested loop. And so this is how you would set migration in SLiM. So you're using the set migration rates method on the subpopulation class. So here we are just indexing this vector, which contains all of the instances of all of your subpopulations in it. And then we are saying we want migrants, and how it works here is we want migrants coming from subpopulation J going into subpopulation I. So we define subpopulations as just a numeric range from 1 to 5 here. So that's why we can call our subpopulation as J. But if you called it like a letter or you called it an actual name, that's where you would call the name. And then this is the rate at which we want it to deliver migrants. So this is not a reciprocal exchange. That's probably also important to mention. It is just a delivery of 5% of your migrants from J into I. So that's how you do that. Any questions? It's kind of a basic, yeah. And then you would just insert your late event because you need a way to end the simulation, and we want our simulation to last 10,000 generations. And we want it to, let's say, output all of our fixed mutations. So this is an example of another version of an output that we can have, is output every mutation. Yeah. Oh, okay. Output every mutation that went to fixation during our simulation. Yeah. Uh-huh. Yeah, so there is a way that you can initialize the tracking of explicit nucleotides and not just like empty bins for mutations. And so then you can modify mutation rates. I believe that's a good question. But I would imagine the point of doing that is so that you can explicitly change or like dictate which mutations, like if it's A to T or something, like a mutation type is always like one to another. So yeah, there is a way that you can initialize that. That would happen in the initialization block. And then you would like set the rates for all of those things. Yeah, I don't know the exact methods for that. Yeah. Okay, so if we run this, we see the creation of all of our subpopulations going. So we have information about all of them here. And then we also see each one on the top panel. And again, the squares are individuals. So once the fitness starts changing, you can see some interesting dynamics start to pop up here where certain ones change color from like yellow to red or green. And then here again are all of the mutations that occurred in the simulation. All right, so this one is, I don't know if this will be quick, but basically I want to show you how to initialize sex and dictate sex ratios in SLiM because that's often a relevant thing to do. Say you want like a different ratio or you want different like age dynamics for your individuals or something, but you need them to be separate sexes. So this is how you initialize sex. So this tells SLiM that you want males and females. And A here means you want to model an autosome. There are also specialized chapters for modeling X and Y chromosomes, or like the sex chromosomes, but I'm not going to get into that in detail here. But this is how you do it. So let's say we want males and females now to occur, and we want them to, this chromosome to model an autosome. And then this is the only other change from before. Basically, when you are initializing your subpopulation, you can also, as a third flag, specify the ratios of sexes that you want. So obviously this would throw an error if you don't originally initialize your sex. But this is the sex of, or your ratio of males in your subpopulation. So this would be 60% males, 40% females. All right, and then that's simulation finished. I think I'll show you the output because there's one thing in the GUI that is worth noting, I think. Yeah, so now you have numbers. Oh, wait, no. Never mind. I thought numbers would occur. Oh, this is the sex ratio up here. And then presumably if there were dynamics occurring, I don't actually know what these do. There's too many buttons on this GUI, but yeah, you're tracking sexes now and you can output different things. And you can also change your output like samples and specify that you want to output females versus males and things like that in different flags of those methods. But okay, so I won't go through this one in great detail, but I'll just show you what the code looks like. So basically this is if you wanted to model a population that works by selfing. So self-fertilization where you're generating two gametes, we all know what selfing is. And then also cloning, so you're generating identical copies of yourself. So this is the only thing that you would have to change. So you add your subpopulation, but then you add a method that's called on your subpopulation class, which is called set selfing rate. And so this 0.8 just means that selfing should be used to generate 80% of your offspring. And then 20% is generated the normal way by choosing a random mate and reproducing. And then set cloning rate works the same way. 10% of your new offspring are produced clonally. Okay, so that's that. So let's get into some actual evolutionary dynamics here, and I'll show you how to model a selective sweep. So something important to mention here is that in order to properly model the dynamics of a selective sweep, you can't specify that one particular mutation will go to fixation. You can't dictate the SLiM that you want this instance of, let's say, M2 to be fixed, because if you dictate that, then that changes the random dynamics that occur in your simulation. And so it wouldn't accurately reflect the biology of what's happening as selective sweep. So you have a few options if you want to look at the dynamics of a selective sweep. You basically can just run a normal simulation while introducing adaptive mutations. So this is an example of how to introduce a super beneficial adaptive mutation. The selection coefficient here is 0.5, which is like crazy high. But basically you could do that. And then after you're done in your output, you could just observe which ones went to fixation and then observe kind of like the structure on the chromosome, look at LD, things like that. In that case, you can't specify where the mutation is on your chromosome because you're basically going in after the fact and just looking at which one's fixed. And obviously also you can't specify when that mutation popped up, right? Because you're just basically allowing the dynamics to unfold and then going in after and looking at your sweeps. If you did want to model an explicit mutation that occurs at a particular time or in a particular location on your chromosome, you also can model a sweep that way, but what you basically have to do is you introduce that mutation in a single randomly sampled individual, and then you just run your simulation a bunch of times and wait for one to occur where that mutation fixes, right? So you're just like replaying evolution over and over again and only choosing the scenario in which that mutation becomes fixed. So there's a way to kind of do this in your code without having to manually go in and restart it every time. And I'll show you how to do that. So this is just a basic early chunk. Here we're defining, we're setting our seed. So what this will do is, we all know what a seed does, but basically it'll set a new seed every time you have to restart your simulation so that the dynamics will change each time and you won't get the exact same outcome. You use the function getSeed to do that, and that generates a particular seed for the particular run that you're doing. And then what this does is basically outputting everything that has happened before generation 1000. So let's say at 1000 is when we want to place our mutation somewhere on our chromosome, because something interesting happens then. So what we want to do is save our output that happened until then so that we can always go back and restart our simulation at generation 1000 instead of having to restart at 0. So we basically save our output to some file, right? And so this will go wherever you want it to go. And then in this block as well is where we're adding our adaptive mutation. So if we want to add a single mutation and we don't want mutations just to occur randomly on our element, this is how we do it. So this is placing a single mutation. That mutation is of type M2 because we want it to be an adaptive mutation, right? And so we are introducing it into this randomly sampled target individual. You can also place a mutation on, say, like 10 or 100 or however many individuals you want at one time, but that wouldn't really be real evolutionary dynamics. You can do it, but this is how you would accurately model a sweep, is sampling one and then placing it using the add-drawn mutation method at this location. And then this is where you are rerunning your simulation if things don't work out and your mutation becomes lost. So that will happen a lot of times, especially if your mutation is not very adaptive, right? So I guess I won't go through this line by line, but what this does is it checks that your mutation either got lost or it was converted to a substitution. So that's why you're counting the amount of mutations of this type, and that if it equals 0, then one of those two things happened. So then you do your checks basically, and if it fixed, you end your simulation early. But if it didn't, you restart. If it got lost, you restart your simulation. And so this is how you would restart it at generation 1000. You use a method called read from population file, read in that file that you created earlier in your simulation, And then you set a new seed because you want different dynamics to occur now after generation 1000. And then you redraw your sweep mutation and you do it all again. So one other thing to note too is the numbers next to late here intuitively indicate that this event should start at 1000 and occur every single generation up until the end of your simulation. So that's how you would induce like a range like this. So if you run this, we see that it was fixed almost immediately and we ended, okay, it was lost and it restarted and then it became fixed on the second run. So if you change your selection coefficient to be like much less, you'll probably see that it will take longer for it to become fixed. Yeah, so it got lost a ton of times before it was eventually fixed. So this is how you would model the dynamics of a selection sweep. Okay, so we only have a few more minutes. I could go through another scenario. I could open the floor to questions, whatever you guys want. Go through. Yeah. Yeah, it got converted to substitution now. So the fitnesses get reset because now it doesn't affect your fitness at all. Yeah, it's good. Yeah, so I have a bunch of examples here of fancy things you can do. Basically, when in doubt, just go to the manual and they have really nicely laid out chapters, which are all links and bring you right to the section in the manual. And then there's always really helpful and nice descriptions of how to think about the code that they give you and how to model stuff on your own if you wanted to tweak the code that's available there. But again, you can also access the code in the GUI by just opening the recipe for it, which is kind of an easy way to do things. But yeah, I mean, maybe I was gonna go through like non-Wright Fisher examples, but maybe we're kind of out of time here. Yeah, the main difference is like you've laid things out a bit differently. And in non-Wright Fisher contexts, you explicitly have to deal with reproduction on your own. So you're no longer choosing a random mate from your subpopulation, which is what happens in Wright Fisher. You have explicit control over which mate you choose. And so in this, what's called a callback, which you have to define, a reproduction callback loops through every single individual in your P1 population, and then you have to choose basically your mate. So you can randomly choose that, or you can introduce fun dynamics like spatial dynamics, where you want mate choice to be influenced by its proximity on your 2D plane, or a lot of other things that you can do. But essentially, you can control like what litter size you have. So this is how you could run into issues where now your population is growing exponentially and you're killing your processor because you could specify your litter size to be whatever you want, right? So if you have it be 1, then your population will quickly go to extinction. So there are kind of like different ways to deal with this and make sure that your size remains controlled. You can calculate what your litter size should be based on various different types of growth models. You can also specify your fitness values and do fitness scaling in non-Wright Fisher contexts using growth models as well. So fitness scaling is like the easy and most intuitive way to control population sizes in non-Wright Fisher contexts. And it's, where is fitness? Yeah, fitness scaling. So this is what it looks like. This is bare bones what you minimally would have to do to control your population size in a non-Wright Fisher case. So in every generation at some point, you should be scaling your fitness in some way to be some proportion of how many new individuals you have and what your carrying capacity should be in your simulation. So that would be K here. So this is how you would do it. Basically, this, as the name suggests, scales everybody's fitness according to some ratio that is a function of what you want your carrying capacity to be and how many individuals you have in your current simulation. And then this basically just controls the distribution and shifts it whatever way so that the appropriate number of individuals get killed off every generation to maintain a particular size. Okay, we're at time, so yeah, I'll end there, but if you have questions, feel free to ask. Thanks. Yeah, so I use it, I'm modeling like a unicolonial population. This is what it looks like. It's like extensive, and basically I'm introducing like a spatial component into it where I'm trying to like model how like a queen's fitness that's indirect gets influenced by the fitness of the other individuals in like a spatial region surrounding that queen, because I'm trying to model kin selection dynamics. So in my case, like I'm modeling sterile workers, and you can't model non-reproductive organisms in SLiM. So I'm trying to like use loopholes and basically influence the queen's fitness indirectly by the composition of basically mutations surrounding the queen in like a nest structure. And then I'm varying like things like dispersal rate and like density and things like that to try to see if that influences like the dynamics of how indirect traits are passed on. If that makes sense. Yeah, it's kind of complicated. Yeah. Yeah.
And when you're making this there's I know that I use and start and then I do that.
Oh yeah. Like I'm not sure about migration in particular, but I know that there's like plotting outputs here in the GUI. So you can plot all of these different things, the site frequency spectrum, the mutation trajectories, fitness over time, like age distributions if you're doing age. So there are like some things built in. Yeah. Oh, there you go. Yeah. So there you go. Yeah, if you want a plot of it. I also didn't discover this function until I was making this presentation. So I've had to do everything manually, but yeah. The GUI has a lot in it. So if you just spend some time learning it, it will pay dividends for sure. Yeah. Any other questions? Yeah, I mean, I run everything on Savio, so there's a command line version of it. And so you just can install the software and build it from the source on Savio. And then you can pass in a bunch of things with different flags, and you just pass in all of your appropriate stuff. But then you just refer to like a script that you wrote, and it will just run it. I don't know how to do threading, and if that's a possibility. I've never looked into it, but you can run it on Savio. It's fine. All right, cool. Well, thanks all for sitting here and listening to that. Got something out of it.